Very good morning, students. So, we have completed the chapter 8 and we are going to start the new chapter application of biotechnology. So, in this chapter, we are going to discuss about how the biotechnology is very much useful in the field of medicine, gene therapy, stem cell therapy, cloning, etc. Various uses of biotechnology we are going to discuss under this chapter. So we already know the definition of biotechnology. What is the meaning of biotechnology? Biotechnology is using some of the biological components with the application of some techniques that can be used for the various purposes in the field of medicine, agriculture, etc. So that is only the term biotechnology. And also in the previous chapter we have discussed about how the microorganisms are involved in the production of some of the products. But in this chapter, how the biotechnology is act as an important tool for the production of many number of products. So that only we are going to discuss. So the first topic what we are going to discuss, how the biotechnology is involved in the medicine field. Especially we are going to learn the three important medicinally important products, how the biotechnology is involved in the production of such products like human insulin, vaccine and interferon. These three things we are going to discuss under the application of biotechnology in the medicine. The first thing we are going to discuss is the recombinant human insulin. You know very well, the insulin is naturally produced by our body, isn't it? Especially one of the important exocrine as well as the endocrine organ in our body, that is a pancreas. We know about the pancreas, isn't it? Pancreas is involved in the exocrine as well as the endocrine function. Exocrine means they are producing some of the enzymes for the digestion. Endocrine means they are producing some hormones. So in the endocrine part of the pancreas, it is produce one important hormone called the insulin. Okay. Usually, talking about the structure of the insulin which is contain 51 amino acids. The insulins are made by 51 amino acids and contain two polypeptide chain. All the 51 amino acids are present in that two polypeptide chain. What is the name of the polypeptide chain? Polypeptide chain A and polypeptide chain B. Here the polypeptide chain A contain 21 amino acids while the polypeptide chain B which contain 30 amino acids. So these two polypeptide chains only together form the insulin. So what is the role of the insulin? The insulin is controls the levels of glucose in the blood. Whatever food we are taking, especially when we are taking more amount of carbohydrate, that carbohydrates are converted into simple sugar. Complex sugars are converted into simple sugar called the glucose. When the glucose is actually the glucose is very important for the body for the many functions and many vital activities. But when the glucose levels are becomes more, that is only leads to the condition called the diabetes mellitus. So that insulin levels are controlled with the help of that glucose levels are controlled with the help of the insulin. So what is the role of the insulin here? It facilitates the cellular uptake and utilization of the glucose. The glucose is not directly reduced, but the insulin makes other cells to consume more amount of insulin for the function. So by that way, the level of glucose is reduced with the help of the insulin. But when the insulin is deficient, I already told you The name of the disease called the diabetes mellitus. So, when the insulin is reduced in the particular person, you have to follow the strict diet plan, and also he wants to take the insulin as an injection. Since their body is not have the sufficient capacity to produce the insulin, they have to take the insulin from the outside through the injection. Then how that insulin is produced? The insulin which is taken through the injection should be produced in the laboratory, isn't it? But how it is produced? In the early years, in the early years, the insulins are isolated from the pancreas of the pigs and cows. But taking the insulin from these animals like pigs and cows have some small difference in the structure. The structure of the insulin is somewhat differ in these animals, including the pigs and cows. That's why when that particular insulins are introduced into the particular individual, the individuals are getting the problems of allergic reaction because it is the insulin of the animals. That's why with the help of the biotechnological tools, the insulins are started to produce from the cells of the human being by the technique called the recombinant DNA technology. So recombinant DNA technology பற்றி நம்ம ஆல்ரெடி படிச்சிருப்போம் இல்லையா? So what is recombinant DNA technology? It is a combination of the two DNA. That is only called the rDNA technology. So with the help of the rDNA technology, the insulins are produced from the human pancreatic cell with the help of the bacteria called the E. coli. So how the particular insulins are produced, that I will explain you with the help of the picture. So before we enter into the picture, what are the important mechanisms that takes place during the production of the human insulin? I already told you the insulins are made by the two polypeptide chain, isn't it? A and B. A and B polypeptide chain. Actually, during the synthesis of the insulin with the help of the rDNA technology, there is another important chains will be linked to the A and B chain. A and B polypeptide chains are linked by the third chain called the C. So, as the C chains are removed with the help of the rDNA technology, the remaining A and B polypeptide chains are together and they involved in the production of the insulin. Okay, only the removal of the third chain C is takes place in the rDNA technology. It is a largest process. So let me explain with the help of the picture how the rDNA is produced. How the insulins are produced. Human insulins are produced. Okay. So for the production of the, I think they are not given the explanation for the production of the insulin with the help of rDNA technology. Anyway, I will explain with the help of the picture. First, what they are doing during the insulin production, they are taking the human pancreatic cell, because the human pancreatic cells are contain the two group of cells, alpha cell and beta cell. The beta cells are contain the beta cells are responsible for the production of insulin. So first what they are doing, they are taking the human pancreatic cell. This is the first step. After that they are taking the bacteria. The name of the bacteria is called the E. coli. So the E. coli bacteria is taken. So first what they are doing, they are taking the DNA from the pancreatic cell and they are cutted the DNA. You know very well how the DNAs are cutted. The DNA can be cut with the help of the restriction enzyme, isn't it? So the DNA from the pancreatic cells are taken and they are cutted into small pieces. Okay, you can see the orange color, no? That is called the pancreatic DNA, off segment of the pancreatic DNA. Again, from the bacteria, there will be a plasmid. You can see here, no? The plasmid. They are taking the plasmid and they are cutted with the restriction enzyme. Restriction enzyme is என்ன பண்றாங்க? That plasmids are cutted. So, the small segment of the plasmid, you can see here, and the small segment of the pancreatic DNA. Both are joined together here. You can see here the two DNA, the DNA from the pancreatic cell and the DNA from the bacterium are joined together with the help of the DNA ligase. DNA ligase is nothing, it is used to join the two segments of the DNA. So இப்போ தான் வந்து இதுக்கு பேர் தான் RDNA, because I already told you RDNA means the DNA which contain the DNA from the two different organisms. That's why they are called the RDNA. So once the RDNAs are produced, once the RDNAs are produced, that RDNA, the single RDNA is not enough for produce the insulin. So it should be introduced into the E. coli bacteria again. Another E. coli bacteria they are taken, then they are introduced the RDNA into the E. coli bacteria. Once the RDNA is introduced into the E. coli bacteria are started to multiply. You can see here. One E. coli bacteria will be divided into 1, 2, 3, 4. For example, they are given 4. But actually, more number of copies of E. coli will be produced. Okay, once the E. coli bacteria are produced enormous copies, they are started to produce human insulin. You can see here the RDNA, which is dot the dot is indicate the production of the human insulin. எங்கே நடக்கும் இந்த human insulin உடைய productionனா fermentation. நம்ம last classல last chapterல we have discussed about the fermentation, isn't it? Fermentor is a place where the fermentation takes place. So here the human insulin production takes place in the fermentation tank. So this is only about the production of human insulin. So we have discussed how the human insulins are produced. Usually the insulin which is produced with the bacteria called the humulin. They are called as the humulin. In the picture அவங்க என்ன கொடுத்துருக்காங்க, I told you, 3 chains will be there. By removing the C chain, இந்த இடத்துல நீங்கள் பார்த்தீங்கன்னா உங்களுக்கு தெரியும் A chain and B chain which is united by the C chain இல்லையா. So by removing the C chain, the two polypeptide chain A chain and B chains are responsible for the production of the human insulin. So this is only the procedure how the human insulins are produced in the laboratory. So that's all about the human insulin production. And now we move on to the, here it is the oldest PDF. So they are given some other medicinally important products, but it is not in your textbook. So we can directly move on to the second products which we can get with the help of the biotechnology, that is the interferons. So interferons are nothing, it is a proteinaceous antiviral substances. Usually பார்த்தீங்கன்னா the interferons can easily produced by our body. When the virus are enter into our body, the cells of our bodies are produce an important proteinaceous substance that is only called as the interferon. But when the interferons are artificially induced to produce in the laboratory, that is only called as the artificial interferon. So interferons are produced in the laboratory. How the interferons are produced in the laboratory with the help of the biotechnology, that only we are going to discuss here. Okay, so interferons are first discovered by the scientists called the Alec Isaacs and Jean Lederman in the year 1957. So that is about the discovery. Based on the structure of interferons is proteinaceous substance, I already told you, no. So when talking about the chemical structure of the interferons, they are classified into alpha, beta and lambda interferon. இந்த மாதிரி நிறைய different types of interferons வந்து இருக்கு. So whatever may be, what is the role of the interferon? The interferons are stimulate the DNA. இதோடைய DNA ஒரு செல்லுக்குள்ளே இருக்கக்கூடிய டிஎன்ஏவை ஸ்டிமுலேட் பண்ணி they produce the antiviral enzyme. that antiviral enzyme which inhibit the replication of virus. That's why we can say the interferons are very much against the virus. Okay. But taking the interferon from the blood, யூஸ்வலாக இன்டர்ஃபரான்ஸ் வந்து எங்கேருந்து எடுப்பாங்கன்னு பார்த்தீங்கன்னா பிளட் செல்ஸ்லேருந்து எடுப்பாங்க. பட் ஃபார் டேக்கிங் த இன்டர்ஃபரான் ஃப்ரம் த பிளட் எனாமஸ் அமௌண்ட் ஆஃப் பிளட்ஸ் ஆர் டேக்கன். நிறைய பிளட் வந்து தேவை இன்டர்ஃபரான்ஸ் வந்து எடுக்குது. தட் இஸ் நாட் ப்ராக்டிக்கலி பாசிபிள். ஸோ டு ஓவர் கம் திஸ் ப்ராப்ளம், முதல்ல ட்ரெடிஷ்னலாக வந்து என்ன பண்ணிகிட்டு இருந்தாங்கன்னா பிளட்டில் இருந்தால் எடுத்துகிட்டு இருந்தாங்க. பட் அது வந்து சரியாக செட் ஆகலை. ஏன்னா வந்து மோர் அமௌண்ட் ஆஃப் பிளட் வாஸ் ரிக்வயர்ட். தட்ஸ் வை தட் த ப்ராக்டிஸ் வாஸ் கேன்சல். பட் nowadays with the help of the rDNA technology, நம்ம படித்தோம் இல்லையா இப்போது, with the help of the rDNA technology the interferons are produced. So the name of the organisms which is involved in the production of the interferons are called the yeast, one of the fungi called Saccharomyces cerevisiae. This is one of the most important microorganism to produce the recombinant interferon when compared with the E. coli. E. coli compare பண்ணும்போது இந்த ஃபங்கையை வச்சு என்ன பண்ணுறாங்க, மோர் அமௌண்ட் ஆஃப் இன்டர்ஃபரான்ஸ் தே ஆர் ப்ரொடியூஸ். பிகாஸ் இந்த ஈ கோலை ஏன் வந்து சரியாக நடக்கலை, ஒய் த இன்டர்ஃபரான்ஸ் ஆர் நாட் டேக்கன் ஃப்ரம் த ஈ கோலை அப்படின்னு பார்த்தீங்கன்னா, த ப்ராசஸ் ஆஃப் glycosylation இஸ் நாட் டேக்ஸ் பிளேஸ் in the E. coli. It is a process of the glycosylation takes place only in that Saccharomyces cerevisiae. That's why they are selecting that fungi for the production of the recombinant interferons. So what is the uses of the interferons? Interferons are used to treat various types of diseases, especially viral diseases like AIDS. அப்போ கேன்சர் வருது, இங்கே கேன்சர் வந்து வைரஸ் மூலமாக கூட சில கேன்சர் ஃபார்ம் ஆகுது, சரியா? ஸோ தட் டைப் ஆஃப் கேன்சர், எய்ட்ஸ், மல்டிபிள் ஸ்க்ளீரோசிஸ், ஹெப்பாடைட்டிஸ், ஒன் ஆஃப் த மோஸ்ட் ட்ரெட்ஃபுல் வைரல் டிசீசஸ், எஃப் ஜோஸ்டர். ஸோ மெனி நம்பர் ஆஃப் வைரல் டிசீஸ் ஆர் ட்ரீட்டட் வித் ஹெல்ப் ஆஃப் this particular interferons. Okay, so that's all about the interferons. So two medicinally important products with the help of the biotechnology we have discussed, and the last product what we are going to discuss is the vaccine. I think you know very well about the vaccines. What is the uses of vaccine? Vaccines are induce the immunity in the human being. Yeah, immunity வந்து அதிகப்படுத்துது vaccine. So from our birth onwards we are receiving the vaccines, isn't it? The vaccines are against various types of diseases like TB, then what we can say hepatitis. Nowadays the what we can say the most dangerous coronavirus. For that also they have discovered the vaccines, co-vaccines by the Bharat biotechnology department, and Covishield vaccine. Two vaccines are produced in India against that coronavirus. So vaccines are the very much important to improve the immunity in the particular person against the various diseases. So that is only the recombinant vaccines. So wait a minute, அடுத்த டாபிக் போயிடுச்சு அது. Okay, so with the help of the rDNA technology, how they are producing the vaccine? So what is the procedure they are doing in the vaccine? அந்த வேக்சினுடைய ப்ரொடக்ஷன் வந்து இங்கே கொடுத்துருக்காங்க. I will show you, I will explain you with the help of the picture now. So you can see the picture here. Actually the picture is not given in your book. So for your easy understanding, I will explain the production of the recombinant vaccine. Especially one of the example here they are given the HB vaccine, hepatitis B vaccine. எப்படி ப்ரொடியூஸ் பண்ணுறாங்க அப்படின்றது வந்து இங்கே கொடுத்துருக்காங்க. ஸோ ஃபார் தட் ஃபர்ஸ்ட் வாட் they are taking the hepatitis B virus. You know one thing, All the vaccines are contain the particles of the virus. Virus உடைய particles வந்து அந்த வேக்சினில் இருக்கும். Then only they can induce the immunity against a particular virus. சரியா? ஈவன் கோவேக்சின், கோவிஷீல்டு இதிலெல்லாம் பார்த்தீங்கன்னா it is also contain the particles of the coronavirus. So, okay, let's go to the production of the recombinant vaccines here. So, first what they are doing, they are taking the hepatitis B virus and they are taking the E. coli bacteria, as we see in the production of the human insulin. Here also they are taking the same E. coli bacteria. So, first what they are doing, they are taking the DNA and they are cutted the DNA with the help of the restriction enzyme, and you can see the orange color part is the DNA of the hepatitis B virus. And again they are taking the plasmid here. The plasmids are cutted with the restriction enzyme and small part of the plasmids are attached here. So rDNAs are produced. You know very well, rDNA means what? Two different DNAs are joined together. That is only called the rDNA. Once the rDNAs are produced, they will introduce into the E. coli bacteria for the multiplication. அங்கேயும் அதே மாதிரி தான் பார்த்தோம் இல்லையா, இன்சுலின் ப்ரொடக்ஷன்லையும். ஸோ ஹியர் ஆல்சோ the rDNAs are introduced into the recombinant bacterium, and the multiplication you can see here. The multiplication of the particular rDNAs are takes place here in the fermentation tank வந்து நடக்குது. So after the multiplication of many numbers of copies of recombinant bacterium, there is a production of the HB antigen. HB antigen is nothing, it is a vaccine. After production of the HB antigen, it will be purified and they will be keep it as a HB vaccine. Now these vaccines can be given to the people in order to prevent the particular person from the hepatitis B virus. அந்த ஆளுக்கு என்ன வைரஸ் வராது? hepatitis B virus வந்து வராது. So this is a method of production of recombinant HB vaccine. So by this way only they are producing the vaccine. So that only they are given in the paragraph also. So that's all about the production of human vaccines with the help of the rDNA technology. So 3 products we have discussed in this chapter. What are the 3 products? One is the human insulin. Second one we have discussed is the interferons. It is the antiviral capacity. And the third one is the vaccine. So that's all about the 3 important products what we have discussed in the today's class. So by the tomorrow's class, we will learn more about the gene therapy. What is gene therapy? What are the different types of gene therapy? Everything we will discuss by the tomorrow's class. Okay. Thank you.